everyone, I'm Mr. Furlong, and today we're talking about cell transportation. We've talked a lot about all the organelles that are inside of the cell and their functions and all these things that are going on, including chemical reactions. And we know that there's thousands of chemical reactions occurring in each cell every second. New molecules are being made, energy is being used, waste molecules are being produced. Thing is, how do these molecules get in and out of the cell? Well, we know that the cell membrane plays a very important role in this. Remember, the cell membrane is what's called selectively permeable. It allows for certain things in and out, but not others. Here we see a diagram of part of a cell membrane, and there's just a few things that I'd like to label on this so that you have an idea as to what each of these different parts are doing. First of all, we see these round yellow parts, and then there's looks like two little legs coming off of them. Well, that is representing a type of a lipid. Remember, that's a fat called a phospholipid. The round part is representing the glycerol backbone that has a phosphate group attached, and then the two little legs, or tails as they're called, represents the fatty acids that are present. Now, they always form two layers, and so this is referred to as a phospholipid bilayer. We have two layers of those phospholipids. Now, embedded in the cell membrane are proteins. And so here we see a protein right in the middle right here. This is a carrier protein. And so this is going to allow things to get in and out of the cell if they can't make it through the cell membrane. And then you can see these chains of molecules. If you look real closely at these molecules, you notice the shape of them. Yeah, they're hexagon shape, aren't they? And so these are a type of a carbohydrate. These are, because they're attached to a protein, these are called glycoproteins. And these are acting as receptors for the cell. We're not going to worry too much about that. Alright, so here is my diagram of a cell membrane. And so you can see the phospholipids, again, the round part with the two tails, and then this is representing one of those carrier proteins. There's another carrier protein there. So how do things get in and out of the cell? Well, we're going to take a look at several different ways. The first is diffusion. Now, diffusion is simply the movement of solids and gases from areas of high concentration to areas of low concentration. Because this follows some law of physics, no energy is needed for these molecules to move. And so one of the more important molecules is oxygen. So let's draw a whole bunch of oxygen molecules. Now remember, the top half, we're going to say that's the outside, and down below here that's the inside of the cell. And we're only showing a portion of the cell membrane. And just by looking at this, we can see that there's a lot more oxygen outside the cell than there is inside the cell. Because oxygen, of course, is going to end up in the mitochondria, and that's where we get to release the energy from our food and so on. Because there's this difference in concentrations, oxygen can just move from high to low concentrations. It's a very small molecule, so it can just pass in between those large phospholipids. Now, one of the byproducts that the cell makes is carbon dioxide. And so we get a lot of carbon dioxide building up inside the cell. There's not much outside the cell, because as soon as the CO2 gets outside the cell, it gets picked up by the bloodstream and taken away. But this does set up a concentration difference. CO2 is also another small molecule, and simply by diffusion it goes from an area of high concentration to an area of low concentration. No energy needed for this. Now, something similar to diffusion, in fact it's a type of diffusion, is osmosis. Osmosis is the movement of water from areas of high concentration to areas of low concentration. So notice we only had that slight difference. Diffusion, solids and liquids. Osmosis is water. So let's draw some water molecules. And this depends. Sometimes there might be more water on the outside of the cell than inside. Sometimes there might be more on the inside than the outside. Sometimes they're going to be the same. In fact, most of the time they're going to be the same. But in this example, here water is in a greater concentration outside the cell than it is inside the cell. And so by osmosis, water is going to go from a high to low concentration, and it's going to end up inside the cell. Now, a third type of diffusion is called facilitated diffusion. This is the movement of substances from areas of high concentration to areas of low concentration, but it has to go through one of those transport proteins or one of those carrier proteins. One important molecule that we have to have is sugar. So let's draw some sugar molecules. This is glucose. Remember, glucose we need for energy. And certainly we have a lot more glucose outside the cell than inside the cell, because as soon as it gets inside the cell, we use it up. We extract the energy from that. But this is a big molecule. Remember, glucose, C6H12O6, that's 24 atoms. It can't just simply pass through those phospholipids, so it needs some help. And it's going to end up passing through those carrier proteins. And these carrier proteins, though, these are specific. In other words, this particular carrier protein would only allow for glucose to get in. If, say, an amino acid needs to get in, it won't be able to get in through this tunnel. It's going to have to find the tunnel or the carrier protein that allows for amino acids to get in. And so this is how cell membranes can be selectively permeable. A bacteria, for instance, simply can't pass through these carrier proteins and get inside the cell to infect us. They do that some other way. The next type of cell transport is active transport. Now, active transport is almost the exact opposite of facilitated diffusion. Here, substances are going to move from areas of low concentration to high concentration. Facilitated diffusion was high to low. This is low to high. And again, it has to go through a transport protein. It's kind of like the cell's pump, if you will. You might have a sub pump at home in your basement so that if water starts to fill up, it pumps that water out so that you don't have any water in your basement. It stays all out in your yard. Let's take a look at an example of this. Here is some sodium ions. We have a lot of sodium in our bodies. Now, sodium, though, even though it's a small molecule, it can't penetrate through the cell membrane because the cell membrane is slightly positively charged. Recall that opposites attract and like particles repel. So if you got a positive sodium ion, it's not going to be able to get through. But we have special pumps that can pump sodium from one side to another. Now remember, this is active transport. So is the sodium going to end up going into the cell, or is it going to leave the cell? Yeah, it's going to go into the cell, right? It's going to go from an area of low concentration to an area of high concentration. This next type of cell transport, although it has to do with the cell membrane, things are happening a little bit differently. The first is called endocytosis. Now these are going to be for really big molecules. We won't have any special carrier proteins that will allow these molecules to get in or out, but these molecules still need to get inside the cell. So the cell membrane can form and envelop those molecules, forming a vesicle or a bubble, if you will, and bring them inside of the cell. So we say that this is endocytosis. Endo means in, cyto means cell, osis means process. So this is the process of cells taking in these large substances. Now some organisms, like an amoeba, this is how they eat. So let's take a look at this video of an amoeba and a paramecium. Both of these are single-celled organisms. But as you can see, the amoeba is slowly enveloping that paramecium. The paramecium is just kind of checking out what's going on. Maybe is this something that I can eat? And eventually, the amoeba will totally engulf and bring in the paramecium inside of its cell. Then the lysosomes come along and they release into that new vacuole that was formed, and it's going to dissolve the paramecium, and that's how the amoeba gets its food. It's going to diffuse those organic molecules into its cytoplasm. Now just the opposite of this is exocytosis. This is the release of large substances from the cell. Remember that we make a lot of proteins inside of our cells that are used in other places of the body. So these proteins that are made on the ribosomes are going to end up going into the rough ER, into the smooth ER. They're going to travel to the Golgi complex. From there, it's going to bubble off and it's going to travel to the cell membrane. And that little bubble is going to fuse with the cell membrane and release its substances. Let's take a look at how this is happening.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
So those are 6 different ways that the cell can get things in and out. We're going to be doing some activities in class that deal with cell transport, and I'll see you then.